we're going to go next to the owner of perhaps the best mustache in marine biology. It belongs to Dr. Russell Arnott, who's going to be presenting with us today. Russell's here now, that you can see that mustache. It is spectacular. He is an educational director at Incredible Oceans, studied marine science at the National Oceanography Centre in Southampton, and worked as a commercial oceanographer before retraining as a science teacher. So combining his love of the ocean and education, he formed Incredible Oceans, a marine education non-profit organisation. And his research now focuses on the overlap between physics and biology, and he seeks to find out how phytoplankton are influenced by different water environments. Welcome, Russell.
Thank you. All good.
Now, I understand you're going to talk about the variety and shapes and sizes of plankton and the tricks they've got up their sleeves for survival. Not that plankton have sleeves, but if they did, there would be tricks in there. But before we get going, do you have a favourite plankton? And if so, why?
I do have a favourite plankton, and it's behind me, and it forms part of my talk, so I don't want to spoil my talk, the end of it, because I think it's pretty cool. But it kind of, if anyone saw Sally's talk at the beginning of this session, she talks a little bit about a species that's quite closely related to it. So, but mine's cooler.
Oh. Shots fired immediately from Russell on the plankton front. Well, I don't want any spoilers, so I say on with the show and let's hear all about it.
Cool. Right. Okay. I'm going to hope that this clicky thing works. Let's see. So this is it. I'm going to talk about why plankton are different shapes. I really like this picture. This was done by an illustrator in Bristol called Scott Masson, who I work with, and told him all about some of the cool plankton that I've seen. And he made this awesome illustration for me. So that was really cool. So I'm going to kind of start off by saying that I'm not a proper plankton scientist. I'm like a pretend one. Okay, so everyone's like, what? But you have a PhD in this. I'm like, okay, well, I'm going to just kind of let people in on a secret, really. And the secret is that I didn't do any science A levels. Okay, so everyone's like, how did you trick the system? How are you able to do this? So basically, I was able to do this I had maths and geography. So I was able to sneak onto an oceanography course. So I kind of learned about biology accidentally, but it's been really cool because as well as being able to play around on ships, and so I went down to the Southern Ocean. I've also got a lab where I grow plankton and I make 3D printed models of them, which I'll talk about in a bit. And I also get to kind of travel around. And so I did a load of my experiments in these things called Mesocosms, which I'll talk about in a bit later. But the other kind of cool thing about my job is that I also run this organization called Incredible Oceans, which I do alongside my research. So basically we go to festivals and schools all around Europe and the UK, and we do kind of cool talks about the ocean. So if anyone is interested in getting us into the school, you can just go to that website and just give me a buzz and we'll see what we can sort out. But anyway, it's kind of cool. Who doesn't like dressing up as a pirate? So so this is my research, and this was a really interesting thing. So it is, why are plankton all kinds of weird and wonderful shapes? Because in theory, when you are tiny, and you think about most plankton, I say most plankton, plankton that I study, phytoplankton, are about the same thickness as a human hair. So at that level, when you're so tiny, water is like thick, goopy, viscous material. So the idea behind it, when you look at the physics of it, is that it doesn't matter what shape you are, because it doesn't work like normal water. It works like sticky, goopy honey. So there's no benefit to being streamlined. So what scientists do when they try to computer model how plankton behave is they just simplify things, and they say, let's just make it easy. We'll make them all spheres. Some people go crazy and make them like rugby ball shaped. And other people are like, no, maybe we can make some that are cylinders. But then you're like, well, hold on a sec. If there was no benefit to being like a funny shape, then why have we got so many weird and wonderful shapes in the plankton? So we were like, okay, so let's see if we can do an experiment to find this out. And the only other experiment that someone's done about this, and it's a really lovely, really simple, but really fun experiment, is this experiment where they made loads of little plankton models using Fimo clay, and they got like a tube of thick, goopy glycerol, and they drop a cylinder in and time how long it took to sink. And then they'd get a cylinder, sorry, they drop a sphere in, time how long it took to sink. Then they would remold it into the shape of a phytoplankton and see how the shape of it changed, how long it took to sink. And so we get this idea of like drag coefficient. And so it's basically, if you're a parachute shape, you're going to sink slower. And so this provides benefit to plankton, because it means that if you're heavy and you can't swim, and you want to stay in the photic layer where the light is so you can photosynthesize, if you're like a parachute shape, or you've got bits sticking out of you that slow how you sink, you can stay in the top layer for longer and get that sunlight to help you grow. So we were like, okay, so this is really interesting about sinking, but not all of the ocean is nice and calm where things sink. Things can be like really turbulent and really mixed up. So we were like, okay, this is interesting. We could do something quite interesting about this. So essentially what's happening in the ocean, and if you've ever been swimming somewhere really nice and warm, what you get is this top layer, which is nice and warm, and then we have this temperature density gradient, which is called a thermocline. Underneath it, we've got cold water. So you might have encountered this if you go somewhere on holiday and you swim on the surface, it's nice and warm, and then you put your legs down a bit and you're like, oh, it's breathing down there. So if you're a phytoplankton, and you can't even talk about plankton without bringing this guy in. So, and you end up in the top, then you're like, yeah, I'm in the top, and you can get all the sunlight. But if you end up being trapped at the bottom, you're like, mate, I'm at the bottom, I can't grow as well because it's colder and I haven't got as much light. So the one at the top is like, don't worry, because I'm in the top and I can bloom and I can go crazy. And then they actually end up blooming in the top and stopping light coming through even more to the bottom layer. They do something called overshade it. So we're like, okay, so this is really interesting. And because of climate change, it's making that top layer warmer, which means that that barrier between the two is getting stronger. So if you're a plankton that can get up into the top layer because you can swim up or you can float up into it, then you've got an advantage. Now, the bad news is, again, about climate change, is that we've got millions of species, right, of plankton. But what it is doing is certain areas of the world are becoming calmer and other areas are becoming more mixed. So typically what we get in calm water, the creatures that like that, the groups, are the ones that can float up, which tend to be cyanobacteria. They've got a little air bubble inside them called a vesicle, which helps them float up, or something that can swim, which we call motile. If you swim, you're a motile species, and this tends to be the dinoflagellates. So they really like it when it's nice and calm and they do well. But if it's really stormy or it's mixed, then these groups like it. And these tend to be the ones that are heavy and the ones that can't swim or float. And because what happens is they get mixed up, back up into the top, where they're like, yeah, I'm at the top where the sunlight is, so they can do really well. So what we're seeing because of climate change is actually changing the type of plankton that can live in different areas, which has obviously big impacts for fisheries that can grow there because they form the basis of the food chains. But unfortunately, there's also nasty plankton, and there's 200 species of plankton that are toxic, and they give off these toxins that they're just not very nice. They give off these toxins as a competitive advantage to stop other phytoplankton from growing. And they form these things called harmful algal blooms. And sometimes we call them red tides because they turn the water red. And if you were to go swimming in this, it would make you really itchy all over. And if you swallowed some, it would make you sick. The problem is that if this washes into a place where there's mussels or oysters or fish that you would want to eat, then that toxin builds up inside the organism in a thing called bioaccumulation. And so then if you were to eat that, it can actually paralyze you, and in extreme cases, it can kill you. And the bad news is that climate change is causing these type of plankton blooms to happen more often and more frequently. So we wanted to see what role a shape played in helping plankton do well in these different, whether it was calm or whether it was mixed. So we came up with these crazy ideas of ways of doing it. So we were like, okay, I'm going to get this really cool thing called a confocal microscope, and it lets me scan and do cross sections through different plankton species and then combine them back together inside a computer, so I can build a 3D model of what that plankton looks like. And then I was like, hold on, I've got a 3D model of it on a computer. Can I make a 3D print of that? So I spent ages messing around with 3D printers. And then I was like, okay, what I'm going to do, kind of similar to that experiment where they were dropping the plankton and sinking and timing how long they took to sink. I was like, I could make a tank that could test how they perform under turbulence. So what I was going to do is get a big tank full of goop, and I was going to put a grid at the bottom that shook up and down to make it mixed. I was going to get some cameras to track how my model went. So then what I would do is I put a ball into the tank, and then I'd be like, okay, in comes the ball. Then I turn on the turbulence, wiggle the grid up and down, chugga, 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 chugga. The ball would move around and I'd map where the ball went. Then I'd get the ball, move that out and swap it in for a phytoplankton kind of model of the same mass and same weight, turn on the turbulence again and see how it performed differently because of its shape. I was like, this is a great idea. And then I couldn't get the camera system to work. And then I was having all these problems trying to use the microscope. And then someone else said, I tell you what, let's just do it with living plankton. And so they were like, there's this awesome facility up in Sweden in a place called Umeå, which is the northern Sweden. And I had to go to this village called Nordbun. This is what it looked like there. It was really cold. And inside it, they had these awesome tanks called Mesocosms. And these are massive. They're like 5 meters high. So each one holds 2 tons of water, and I could pump them up full of water from the Baltic, and I could set up different mixing conditions inside the tanks to see how the phytoplankton reacted. So I ran this experiment for like 2 weeks, and then I would take samples of phytoplankton at different depths, and then I'd use the coolest machine ever, right? It's this thing called a flow cam. And like I said before at the beginning of my talk, I'm not a proper plankton biologist. I never did biology. Don't tell the Marine Biological Association, okay? So I never did this, right? So I was like, well, I don't know how to identify different plankton. And so I was there with a microscope, and it was taking me ages. And then someone from the Marine Biological Association said, have you seen this thing called a flow cam? What you can do is you can just pour your seawater in the top, and every time a plankton cell cuts a laser, it takes a photograph of it. So whereas this is it happening live, so whereas I was counting them, I could do 300 in like an hour and a half. This thing, I could photograph 6,000 plankton cells in 10 minutes. I was like, what? This is crazy. And then you build up these big image libraries on your computer of all these different plankton cells. You can sort them all out. So whereas other people have got like nice photos of their holiday and stuff on their computer, on my laptop, I have got, and I'm so proud of this, 462,000 images of phytoplankton. And because I've got all of these things and they're digital thing, I can analyze them to see like, okay, did the round ones do well? Did the long ones do well? Did the ones that could swim do well? Did the ones that could float do well? And so I was able to go through and kind of tease that apart. And it was a really cool experiment. So again, I showed you at the beginning of my talk, that illustrator, Scott Masson. He was really excited about my research. He actually made this picture with the Mesocosms and some of the phytoplankton species that I saw in there, which was really cool. So thought I'd talk a little bit about some of the species. Now, as I said before, the funny thing about being someone that knows about physics, but doesn't really know anything about biology, but I have to pretend that I know things about biology, is I had all these photographs and I didn't know what the names of them were for ages. So I was like, okay, now I know they're called Periduniella and Pauliella and things like that. But at the time, I was like, well, I don't know what these things are, but I need to start until I can send the pictures to a proper plankton taxonomist who can tell me what these are, because, you know, it wouldn't stand up to scientific scrutiny, me doing it. So I called them things like centipedes and microchips and DNA and canoes, things like that, which I thought was, and so me and my supervisors, also physicists, would be like, well, it looks like the canoes are going up, but the microchips are going down and things. So it was, I think what I like about this is there's this idea that scientists know everything about everything, and you don't at all. And you kind of have to make it up a little bit as you go along. So I know a lot about these species, the species I study, but if I'm honest, I don't really know that much about other species outside. I kind of look at how phytoplankton on the whole behave. So I thought I'd show my favorite plankton ever, and this is also one of the ones that I had. And this plankton just blew my mind as a kind of physicist that didn't know anything about plankton. Okay, so it's called Mesodinium rubrum. And from the top, it looks like a sunflower. I was like, that's cool when I was looking at it under the microscope. But then from the side, it looks like this. And it's basically two balls on top of each other. And so what it does when it's just hanging out in the water, it's got 6 arms that it kind of pokes out like that. And that stops it from sinking. So it kind of increases its surface area to stop it dragging. If it feels something swim towards it, it can detect the pressure wave using that belt, that tutu that goes around the middle of it between the 2 balls. And then so it detects it. Then what it does is it wraps those 6 tentacles around the top half, and then it just flaps its tutu and it can jump. Okay. So bear in mind, this is something that is the thickness of 1 to 2 human hairs. And what this can do is it can jump 1.2 centimeters. Okay, so to scale that up, that is like us being able to jump up in the air and like high five an aeroplane that's jumping overhead. And you think about that we live in, air. This thing lives in water, which for it is as thick as treacle. So I was just like, this is absolutely mental. And then as I started looking into it more, I was like, what's this thing down the bottom here? That is a mouth, right? So when I ran my experiment, I put a filter on so I didn't have any zooplankton coming in and eating my phytoplankton, because I wanted to see how the phytoplankton responded. I didn't want to know about that annoying animal stuff. And what I was not expecting is the phytoplankton to eat each other. So one of the things that make a phytoplankton is they photosynthesize. They can use the sunlight to get energy to survive. When Mesodinium is first created, it doesn't have any chloroplasts, so it can't photosynthesize. So what it does is it eats another cell and steals the chloroplast from inside it. So this is like you being able to eat a lettuce and then absorb the chlorophyll into your skin, so you'd kind of go a bit greener, and then just standing outside in the sunlight and just making your own sugar. I was like, this is crazy. So I'm hoping I've got some videos here of it doing its thing. So here it is, much like Sally's videos. It's just hanging out and it can't see, and it uses that tutu belt to detect a cell swimming near it, and then it's just like, nah, mate, I'm gonna eat you. And then this one again, there should be another video come up now, and I'm hoping, here we go. So this one, the stupid cell hasn't realized that it's going around. As soon as it hits, boom, there we go. It touched that tutu, and Mesodinium ate it. But again, before we knew that this was Mesodinium, we knew that it was eating things, and we thought they looked like sunflowers. So for ages, we just call these chompy suns. And so I still call these chompy suns. So on my bucket list is to get a Mesodinium tattoo. I have yet to do that. So there we go. So that's kind of cool. Thought I'd end just with a shameless plug. Christmas is coming up. And if anyone needs an awesome Christmas present, there's this book that I wrote that's just full of really pretty pictures and loads of cool stuff. And obviously, I try to talk about plankton as much as I possibly can in it. There we go. But anyway, I'm going to stop waffling on now. And this has been me. And I'm going to do some questions. And if anyone wants to get in touch with me after this, there's my email address. And you can get in touch with the website as well. But thank you.
Fascinating stuff, Russell. Thank you. And before we get into some questions, a reminder to everyone that the hashtag for this is YMB Summit 22 on Twitter and other social media. So get involved and don't forget to tag at MBA UK as well. And Russell, delighted to see plankton, my old friend from SpongeBob, cropping up there. I've always wondered what species plankton actually is. They never quite define it.
He's a copepod because not many people know that Stephen Hillenburg, creator of SpongeBob, was actually a marine biologist before he did that. So plankton is actually a copepod.
Love it. Love
There you
it.
go.
I think we've got time for one question, Russell, and the one I'm going to select is this question we got earlier, which was, do some plankton need different nutrients to survive?
Yes, so just the same way that all plants that grow on land need slightly different nutrients. Some need phosphorus and phosphates, and some need nitrogens, and some need silicon. So diatoms, which we'd heard about, they make this like big skeleton, big heavy skeleton out of pure silica. So they need silica to grow. So if there's a place where they don't have any silicon, diatoms don't do very well. Mesodinium, for example, that really, really likes phosphate. Most plankton need like a mixture of all of these. And what's quite interesting, there's actually a micronutrient, which is iron, which is needed in a lot of photosynthesis. So this took scientists a little while to realize, because they would go to the middle of the Pacific and they were like, well, hold on, we're measuring really high levels of all of these nutrients. So why haven't we got any phytoplankton growing here? And the reason is you need iron as a micronutrient. So there's places where you get big plankton blooms. One of the places is off the west coast of Africa. And there's plankton bloom there that's caused by Saharan desert sand, which is really high in iron, which gives it that red color. And that Saharan desert sand blows into the seawater there and causes phytoplankton to bloom. So there's one kind of school of thought that I'm not sure how scientific it is, but there's certain evidence that's pointing to this, that whale poo is really, really high in iron. So what whales do is they take iron from one area, and as they go around and swim around the ocean, they poop en route and effectively fertilize the ocean. So there's one school of thinking that potentially, because we hunted whales so aggressively in the last century, that have we reduced the primary productivity in the ocean by reducing the amount of iron in it? But there we go.
Fascinating. And again, Russell, a fulsome answer. I would expect nothing less. A brilliant presentation and always full of insight. Loved it and beautifully presented. And you're going to be back for our careers panel as well. So thank you very much and look forward to seeing you there.